Branching off from that central spine is the peripheral network, a web of nerves extending to the body's furthest extremities. This architecture divides the labor. The peripheral network operates as a data-gathering web, while the central hub handles the processing. The foundational hardware unit powering this entire network is the nerve cell, or neuron. The human brain alone contains approximately 100 billion of these individual nodes. To function as a network, these nodes must communicate across the physical distances within the body. Every detection of light, sound, pressure, or heat relies on this specific hardware routing data to the appropriate center. Human movement and reaction are the direct results of this parallel processing biological architecture. To understand how the network functions, we have to scale down and isolate the anatomy of a single neural unit. In this schematic, the core processor is the cell body, which houses the nucleus. Connected to this core are specialized biological receivers called dendrites, designed to accept incoming data. Extending away from the cell body are the axons. These act as the transmission cables, carrying output signals away from the processor. Because they connect distant regions of the body, these axons must span relatively large microscopic distances. To see how this unit converts an external stimulus into a transmittable signal, we can trace a single unbroken signal path. Routing a pain signal from the hand to the brain for conscious processing introduces latency. In a moment of physical danger, that delay leads to tissue damage. The evolutionary solution is the reflex arc, a hard-coded, localized circuit designed for immediate execution. The circuit activates when a receptor in the skin detects a sudden change in temperature. In this cross-section, a sensory neuron picks up that thermal data and routes an electrical impulse inward, away from the skin. Instead of traveling to the brain, the signal travels directly into the spinal cord. Inside the spine, a relay neuron acts as a bridge, instantly rerouting the incoming signal. The relay passes the data to a motor neuron, a command pathway that carries the executable action back toward the extremity. The impulse hits an effector, forcing the muscle to contract and pull the hand away from the heat source. The circuit relies on a specific functional taxonomy, the sensory neuron as the detector, the relay as the router, and motor neuron as the actuator. By bypassing the brain, the reflex arc trades complex thought for the raw survival utility of immediate mechanical actuation. While helpful to trace a continuous signal, the network is composed of discrete units. The neurons do not physically touch. Between every cellular unit is a junction point, a physical gap known as a synapse. A subset of synapses are strictly electrical, allowing ions to flow directly between cells, but these are rare. Most junctions in the human nervous system are chemical. This close-up illustrates the primary physics problem at these chemical junctions. An electrical impulse cannot arc across empty biological space. To bridge this void, the nervous system must translate an electrical voltage into physical matter. This translation begins when the electrical pulse reaches the end of the axon terminal. Embedded in the cell wall are voltage-gated calcium channels. These detect the arriving electrical spike and immediately open. Outside the neuron, calcium ions sit at a higher concentration than on the inside. Driven by this gradient, the calcium rushes inside the cell. Inside the terminal are synaptic vesicles, storage containers holding a chemical payload. The influx of calcium forces these vesicles to migrate toward the membrane edge. Upon contact, the vesicles merge their biological material with the cell's outer membrane. This fusion allows the neurotransmitters to be dumped directly into the empty synaptic cleft. In a fraction of a millisecond, raw electrical data has been converted into a localized chemical spray, circumventing the physical gap. Once released, the neurotransmitter molecules drift across the synaptic cleft. Waiting on the membrane of the receiving cell are targeted receptor proteins. The chemical message is accepted only when the neurotransmitter molecule sits into the specific geometric shape of the receptor. A successful docking forces new ion channels to open, letting ambient ions flow into the receiving cell. This ion flow creates one of two binary outcomes. The first is depolarizing, which makes the inside of the cell more positive, continuing the electrical signal. The alternative outcome is hyperpolarizing, which makes the receiving cell more negative, halting the signal. Biological computation relies on this cycle, translating electrical spikes into chemical diffusions and back into precise voltages.